Much, Laura. Hopefully everyone can hear me. I'm just gonna go, and if you can't hear, I guess maybe in the chat, let Laura know. So thanks for the introduction. Laura, I also want to thank the Michigan State University for hosting this webinar. So we're on slide 2. I come from an engineering background. I want to give you some context of kind of where I was coming from and where we're going. And I did my undergrad electrical engineering, my master's in systems engineering. And for my PhD, I wanted to find a topic and a problem area that had a large impact. And I ended up settling on the water industry. And a couple of the main reasons I chose to research water is because I never realized how privileged I was with all the clean water I have. The 663 million you can see on the slide, that's the amount of people around the world every day that don't have access to clean water. And to put that into perspective, that's around 8 times the population of Canada. So I know most of our listeners are from the Great Lakes area, so I think they'll put it into perspective. Even to bring it closer to home, that's 1 in every 10 people. So if you're at a party around 10 to 20 people, that means 1 or 2 people won't even have clean water that day. So providing clean water, knowing if your water is safe and clean to drink, is a very, very important issue, and that's motivated and driving all the research we're doing. Right around the same time that I started my PhD at the University of Waterloo, the Aqua Hacking organization, which is hosted by the Gaspé de Beaupier Foundation in Montreal, put out a competition called United for Lake Erie. And you can see a satellite image of Lake Erie. This is from the MODIS satellite. And in the bottom left, you see a severe algal bloom. And these algae blooms in Lake Erie normally happen every year. We have some good years, some bad years, but they can cause severe side effects. So I'm on slide 4 now. Just for some context, this is what an algae bloom looks like close up. This image was taken in China, in Tahoe Lake. And the water turns all green. This is a severe case. The water will give that stench. It'll have a very, very bad smell. So why are algae blooms such a hot topic right now? The main reason is because different types of algae and cyanobacteria, so we have 3 on the left there of common types, they can produce different types of toxins. So they can produce hepatotoxins causing damage to our liver, neurotoxins causing damage to our brain and our nervous system. And in large quantities and dosages, these toxins can even be fatal. So so for example, our clean water that we have from our water treatment plants, or all the recreational use like swimming or skiing or boating or going to the beach, or also like the tourism components, the fishing industry, all of these are at serious risk when we have these algae blooms taking place in our water, because they can produce these lethal toxins. And these algae blooms are also happening all over the world. So we personally know about the one in Lake Erie quite well. They're also happening in the Mediterranean. I mentioned the one in China. They're happening in Australia. And with climate change and global warming, these algal blooms are also expected to get worse and worse. So the problem isn't going to go away anytime soon, unfortunately. So all of this begs a very big question, which is, how do we know if we have a bloom, and how do we know if we can expect one? And how do we know what types of algae and bacteria are actually in our water? Are they safe algae? Are the algae that will be producing toxins? Are they dangerous algae? So the current process to determine what types of algae and bacteria is in your water is a fairly long process. So first you have to take a water sample, and then you have to transport it to a certified lab. So that can be in a car, a truck, a plane, a train, but you usually have to travel some distance. And then in the certified lab, you'll have a highly trained professional, they're called a taxonomist, and they're specialized in algae and different types of bacteria. And they'll manually look through a microscope and identify all these different organisms. So you can imagine that this is very, very time consuming because you have to take the sample away from its source. And it can be a very tedious process for this taxonomist to do this for a large number of samples. And all of this in turn, of course, costs a lot of money. So when I heard about this problem, when I started my PhD, I thought to myself, there has to be a better way. And of course, not being from the water industry, I thought with my engineering mind. And during my master's, I learned about something called the ImageNet Large-Scale Visual Recognition Competition. And this is a commonly known competition in the machine learning and artificial intelligence world, where you have 20,000 different types of ambiguous categories. So these are all images. You have millions of images, and each image is one of 20,000 categories. So you could have an image of a dog, a cat, a goat, I don't know, a chair, a table, a coffee mug, a computer mouse, like a Sharpie. The list can go on and on and on and on. There's 20,000 of these different types of categories. And just a few years ago, the state-of-the-art machine learning and artificial intelligence, or AI, algorithms were able to achieve 95% accuracy when classifying these different images. And this is a huge accomplishment because when they asked humans to do the same task, it took a lot longer, and on average, they were not able to get the same accuracy performance. So with the computer, they were able to get a 5% error and 95% accuracy, which was higher when testing compared to other humans, and it was much faster. So the main proposal of my PhD and what we're working on is essentially trying to automate the process of algae identification. So we've built a custom microscope system that leverages unique wavelength LEDs. We use off-the-shelf components, scientific cameras, 3D printed material, and we couple that with an artificial intelligence engine, which leverages computer vision, statistical models, machine learning, and different optimization techniques. And then you can take a water sample on site, place it in this instrument, and then on your laptop, you can analyze the results. And our goal, our ultimate goal, is to get the type of algae in the water within a matter of minutes. So we proposed this solution, and we built a prototype for the aqua hacking competition that I mentioned earlier. And we ended up winning first place, which we're very grateful for. So this was a 5-month-long competition. And then we were under the name Sim Labs. We recently changed our name to Blue Lion Labs. And the aqua hacking competition really gave us a lot of exposure to the water industry, a lot of mentors, but also a lot of business mentors. And I also want to say the aqua hacking has another competition for 2018. I had a very positive experience with it. The link is here on the slide as well. And this year they have 4 different problem areas. So maybe one of you on this webinar has experience with one of these 4 problem areas and could maybe start a team. So they're water crisis prediction and adaptation, phosphorus capture and recycling and farming, tackling endocrine disrupting chemicals, and real-time reporting sewage overflow and untreated water spills. So that's kind of a bit of our backstory, where we're coming from, why we're doing what we're doing. Now I want to share with you a bit more of the research side. This is a paper we just finished writing last month, and it's titled The Feasibility of Automated Identification of 6 Algae Types Using Neural Networks and Fluorescence-Based Spectral Morphological Features. So the goal of this paper is essentially to determine how, is it feasible to have an instrument to identify different types of algae. And what we did is we took 6 different types of algae, which you can see there on the left, and we imaged them with our custom imaging system, which generated a number of spectral images. We did some image pre-processing, which resulted in clean images, and then we segmented those clean images to give segmented images. And now that we have segmented and localized different organisms in an image, we extracted different features, which were fluorescence-based, and spectral morphological, which I'll go into more details. And then we compared 3 different types of models to see the relative performance of how well they could classify. And I will also go into that in more detail. And if you want to read the full PDF article, it's at the link provided in the slide. So we're on slide 17 now. So we purchased 6 types of algae from the Canadian Phycological Culture Center, or the CPCC, and they're located at the University of Waterloo. They were from 3 different phyla classes. So the first phyla class is the chlorophyta, or also known as green algae. The second phyla class is the cyanophyta. These are the celebrities of the algae world, or the blue-green algae, or cyanobacteria, and they're the ones producing most of the toxins. And then the third phyla class was the euglenozoa. And within that, we have different species of algae, some from the same genus and some from different genus. So on the top left, we have the Cynodesmus obliquus and the Cynodesmus quadricata. And they're both from the green algae group. And then also the Anchistrodesmus is also from the green algae group. And then on the bottom left, we have the Anabina floss aqua. That's a very, very common type of cyanobacteria. Beside that, we also imaged the pseudoanabina, which is also from the cyanobacteria phyla. And then finally, the last one we have a euglenozoa. So the reason we selected these ones specifically is because the anabina and the pseudoanabina are both filamentous types of algae. And they're very similar looking in nature. So we wanted to evaluate how well this imaging system with our artificial intelligence engine, how well could it separate between these 2 types. And then there's also similar with the Anchistrodesmus, some short anabina sometimes look like Anchistrodesmus. And the euglena and the Cynodesmus can also sometimes look similar. And in this situation, the Cynodesmus obliquus is significantly smaller than the Cynodesmus quadricata. So we extracted for each of these different types of algae spatial features. So essentially trying to describe in ways that we can comprehend how these algae look. So we took the total area, which is the number of pixels in an isolated microorganism, the convex area, which is the total number of pixels of the convex hull. And the convex hull, the best way to quickly describe that is if you were to take like any type of object, let's say like a starfish shape, and you were to put an elastic band around it and let go of the elastic band, the convex hull is the area inside the elastic band that's around your shape. The eccentricity, essentially how, I think that's self-explanatory. It's the distance between the foci of the ellipse and its major and minor axis. The equivalent diameter, it's the diameter of a circle with the same area as the isolated organism. And the extent, which is the ratio of the pixels of the actual organism and to the bounding box that's containing that organism. So we have those 5 spatial features. We also extracted some spectral features. So these are fluorescence-based spectral features where we cause the algae to fluoresce at 6 different wavelengths and then measured the emission intensity. So here you can see the 6 different types of algae with their CPCC number. And I'm just going to go back some slides. This is why the CPC number is included on the actual also algae name for reference. And then on the bottom, you can see the excitation wavelength. So these were created from high power narrowband LEDs. So we have 405 nanometers, 420 nanometers, 450 nanometers, 470 nanometers, 500 nanometers, and 530 nanometers. And just by inspecting these images by themselves, you can see a few things where, for example, the euglena, which is on the bottom, is brightest around 500 nanometers compared to, let's say, the Anchistrodesmus, which is CPCC 366, which seems to be brightest, well, maybe around the same area, but more consistent. The CPCC 067, which is the Anabina, is brightest around 420. And so just getting an intuitive idea of how at different wavelengths these different algae essentially emit different amounts of light when excited at different wavelengths. And the primary reason for this is because of their different pigmentation. They have different, the relative amounts of pigmentation in these different types of algae is different. So the amount of chlorophyll A, the phycocyanin, phycoerythrin, phycoallicyanin, the beta carotene, all of these different pigments. And then we took all these features. This is a very high level of what's called a feed-forward neural network. We took these features and we passed it through a neural network. I won't get into too much detail because this is coming more from a machine learning background. And I want to stay high level and focus on the water aspect of this. But this is just a way to map your features to what different types of algae it is. And what you do when you train this architecture, this feed-forward network, is you show it examples of what one type of algae looks like and the corresponding features that describe it. And you do this repeatedly over and over and over. Essentially, when you're teaching a child how to recognize different types of cars, or you're teaching it new words. And then what we do is we have images that it's never seen before, and we show it to the network, and we ask the network to predict what type of algae it is. And since we know prior what type of algae it is, we can evaluate how well it's performing. So this is the overall architecture again, the system overview, with 3 different types of models. We have model 1, model 2, and model 3, and the relative accuracy. So this was run 15 times with random sampling every time, and always holding out data, test data, to test the network's performance. So model 1 was only trained with the morphological features that I talked about, the 5 different spatial features. Model 2 was trained only with the spectral features, and then we combined all those features together and we trained model 3. So immediately we can see when classifying 6 different types of algae, model 1 has the lowest performance, and model 2 and 3 are significantly much better and very, very high. We did a statistical analysis to see if there's any statistical significance between model 2 and model 3, and we found that there was none at all. But we did from model 2 to model 1, there was a significance, and also for model 3 to model 1. So this study essentially shows that using predetermined spatial features may not be the optimal method when identifying different types of algae, and the spectral features in this situation with 6 different types of algae perform much, much better. In the future, we will also look at automatically extracting spatial features that aren't predetermined by us, but that the machine learning algorithm determines by itself. So right now, during my PhD, we're building the prototype. We're on slide 22. And our goal is to bring this to market. The next step after we're done building our prototype is going to validate the technology, to compare it to other industry standards. And then our goal is to do field testing. So, and the main advantage once the system becomes marketable is it dramatically reduces the cost per sample, and also dramatically reduces the turnaround time. And all of this results in a higher sample throughput. You can essentially analyze more samples in the same amount of time compared to a human taxonomist. And this will essentially allow active monitoring, because now we can have this system which can be brought to the source of water, and let's say every hour take a sample and analyze it. And then the long-term plan essentially is to build up a database of algae activity over time, so we can understand the behavior of algae, especially if we start taking other measurements like pH, or the phosphorus and nitrogen content, the temperature, all these different factors and variables, and seeing how they correlate or relate to the different types of algae, their rate of growth, and what types of algae are present. So, and then the main advantage of this is essentially we can be proactive in seeing when an algae bloom might come opposed to being reactive. Just imagine being a water treatment plant operator and knowing maybe days in advance, let's say, that a bloom might be coming based off of the data and the predictions, opposed to being reactive, where the bloom is happening and you're not sure how bad it's going to get and what's will happen in your water. So we're currently working with a number of organizations, both like water treatment plants. We're working with 2 water treatment plants in the Greater Toronto area. We're working with the Federation of Ontario Colleges Association and also the Sunfish Lake, which are lake associations. And we're also working with the research organization, the St. Lawrence River Institute. And they're essentially helping us test our device and determine the usefulness of it. And we're always looking for more collaborations, more partnerships. I truly believe that I can't accomplish all my research on my own, that I need to collaborate with other people, learn from other people, and find win-win relationships so we can help each other. So this is a quick image of me. That's my name. You can contact me with either of those emails. That's my Waterloo email, and that's my Gmail account. I'm happy to talk to anyone about anything related in this presentation. And that's the end of my presentation. So I guess we can take questions now. Thank you.
Thanks, Jason. Should anyone have any questions, feel free to type those into the chat box. And also, if you wish, you can go ahead and unmute yourselves. We do have one in the chat, and I'll read that off right now. Claire asks, how do you anticipate that the system will perform in distinguishing taxa that have high morphological diversity? Many cyanobacteria occur as groups of cells, colonies, fascioles, filaments. A good example is Microcystis, Anabina, Lemmermanni. Other similar systems, for example, Flowcam, have difficulty recognizing these taxa.
That's a great question. Thanks for asking, Claire. Right now, I anticipate that there will be some challenges there because, well, the morphological diversity, if the whole hypothesis of my PhD is that if a human can recognize the different behavior and morphological diversity within a given species, let's say, well, which is the one, I'm just going to look at the chat. Well, like Microcystis, for example, it can form colonies, or maybe it could stand by itself. If we see enough examples, like if we show the computer, the imaging analysis system, enough examples of what it looks like, then we can teach it the different behaviors. So like right now we haven't explored that. We're primarily like exploring species that are relatively spread apart.
All right, are there any other questions that folks have? I know some of you might be thinking or you might be typing, so we'll go ahead and give you some time to do that.
Yeah, and I just want,
oh, go ahead.
Sorry, I just want to add to Claire, if you're not happy with that answer or you want to talk more, feel free to email me. I'm happy to discuss more.
So once again, go ahead and enter any questions that you have into the chat box. If you are only connected by phone, you can try and unmute your phone and ask your question directly of Jason. Also, if you're on the Zoom, you can go ahead and unmute yourself. Again, if you hover over the bottom of your Zoom window, at the very bottom left-hand corner of your screen, you should see an unmute option. We do have another question in the chat box. This is from Lois. She asks, is there a minimum concentration of a particular taxa that is needed to be detected when run through the imaging system?
Yeah, thanks for the question, Lois. It's also a great question about the limits of detection. The more I spend time with different people in the water industry, microbiologists, this is a common question that has been coming up. And I fully don't know the answer to it right now. There is going to be a minimum concentration, but right now we don't know specifically what that is. So that's something we will need to explore.
All right. Thank you. And we do have another question here. This is from Douglas. He asks, in the real world versus cultures, there are issues with both human and automated identification of algae when there's lots of sediment and detritus. In other words, in western basin of Lake Erie. Are you going to eventually run field samples for your project?
Yes, that is, thanks for asking, Douglas. That is the ultimate goal. So I mentioned we're in the prototyping stage, and eventually our plan is to get to field testing, because it's one thing to build something in the research lab and test it and develop it, but in order for it to work in a real world scenario, these types of tests will need to be done. So we anticipate also with samples with lots of sediment, the fluorescent features would also be very useful because they can separate the chlorophyll containing content of the water sample compared to the other content. And I have read a couple papers where they said that this is a viable method. So that's one thing we plan on exploring too. But I'd love to hear any feedback you have or thoughts on that issue as well.
All right, great questions. Again, keep them coming in the chat box, or you can go ahead and unmute yourself. We do have another one from Frank in the chat box. He is asking a similar question to what Douglas just asked. He says, how accurate is your segmentation step? Might want to explore applications of our CMEIAS color as segmentation tool to help with complex color segmentation problems. Also, have you explored our morphotype classification algorithms for microbial morphological classification using our CMEIAS bioimage informatics software? And he provided a link to that in the chat.
Awesome. Thanks for that, Frank. I'm just reading the question as well. Our segmentation step, it's right now it's based off of computer vision image segmentation algorithms. We have a different types of approaches that we're exploring. I haven't specifically heard of the ones that you mentioned, and I'm definitely going to look into it and check out the link. So thank you so much for providing that.
All right, are there any other questions out there? Looks like we might have one more. Lois asks, some algal species have epiphytes with varying pigments growing on them. Would this produce errors?
It's a great question. Thanks again, Lois. It definitely could produce errors, especially if our algorithm has never seen that scenario before. And that's also where more testing will have to be done. So that's kind of, it definitely needs to be explored.
All right. Again, once again, type any questions that you have in the chat, or you can go ahead and try to unmute yourself. We'll give folks a little bit more time here. And we will send Jason's contact information out along with the recording of the webinar and some of the links that he had provided in his presentation, should you have any other questions following the webinar.
Yeah, absolutely. I'm happy to talk to anyone. So.
Any other questions that folks have? We'll give you another minute just in case you're typing or thinking about something. All right, well, I think at this time we'll probably start wrapping up here. Many thanks to Jason for being willing to present his research, and thank you to everyone for coming and participating in this webinar. Again, we'll send out a recording and some other information that Jason provided in his presentation. You will also receive, and actually I should put that in the chat box, a link to the survey. It's very, very short, so it shouldn't take all that long to fill out. So we'll get you a link to that, and I'll put that in the chat box right now. And you'll also receive it via email.
Great. Well, I also want to thank everyone who attended this webinar. I really appreciate you taking time to listen to me speak and to learn more about my work. I'd love to hear more about what all you are doing as well. And if you have questions, please ask. And Laura, thanks for hosting. I really appreciate it.
No problem. We do have a couple other comments in the chat.
Okay.
Frank, who had asked a question earlier, provided some contact information and more information about his research that he's doing. And Douglas asks if we're able to send the PDF of the talk out too. Yes, you should have received an email with that already, but if you didn't, we will include that with the follow-up email that we send out. So we will get that out to everybody. All right, well with that, I think we will go ahead and conclude the webinar. Thank you again to everyone, and look out for some follow-up emails with additional information on the presentation today.
Okay, thank you everyone.